All right, good evening, everyone, and welcome to Lecture 2, Part 1 of Biostatistics, BIO347. This lecture is going to focus on Neyman-Pearson statistical testing, or the School of Statistical Inference. As I mentioned, I think in the first lecture, statistics is a science, and so it progresses, and certainly not a smooth progression. And with that progression, you have different schools that form that take different paths to understanding data and what it tells us about the world. The two main schools that are is the Bayesian School of Statistical Inference and the Neyman-Pearson School of Statistical Inference. If you ever did a chi-square test or t-test and you look up the critical value on a table with the given degrees of freedom, you did Neyman-Pearson. We also call this the Fisherian significance testing or the Frequentis School of Statistical Inference. Okay. And it's called Fisherian because Sir Ronald Fisher was a statistician, and he came up with the rule of thumb that if your p-value is less than 0.05, you reject the null hypothesis and your data is significant. So that all comes from a guy named Ron Fisher. All right. So these are the things on the left are the things we'll cover. So we've already covered the two schools of statistical inference. We talked about Neyman-Pearson, that it's also called the Fisherian school, or the Fisherian significance, and Frequentis. All right. So how does Neyman-Pearson work? So how do you discover or make statistical inference from data using this approach? First, you do your experiment, and then you calculate something that's called the test statistic. So the tests, so once I tell you some, you'll probably know what these are. So test statistic, right? I'll name a few. All right, so if you calculated a chi-square or a t-test, right? So you get a t-value, okay? If you run a Mann-Whitney-Wilcoxon test, you can get a U, there's a W, there's an F, ... okay? So these are test statistics. Most of these tests are a signal to noise ratio. Okay. And jump ahead just slightly, the noise would be your variance. So that's how much kind of wiggle you have in your system, and the signal would be your effect. So for example, if you were looking at the effect of fertilizer on corn height, a strong effect would be the fertilizer has a greater effect on corn height than the control group, and then the noise would be since your corn in a field would have all these different heights, right? That would be the variance. So the question is that height to height variance among things, is it due to fertilizer or is it due to just random variation in corn heights, right? So that's what most statistical tests are after. So you get and I'll give you an example. So for example, a t-test, right? If you've ever run a t-test, and this is not the formula, okay? This is just the idea of a t-test, okay? Where this is the difference between the means, right? So this is your signal, would be, say, the control field and then the fertilized field. This could be your control mice, and then the mice get a particular drug. But a t-test is just comparing two means. And then the sigma, let's put sigma squared here. This is the variance. So the pooled means across your whole system, okay? So this is not the formula for t-test, but it's there's a little bit of this in the formula for a t-test, but this is like the idea behind a t-test, the signal to noise ratio, okay? So you get your test statistic, right? So you get your, let's say, your chi-square observed, and then you compare to your critical value. So your chi-square critical value, okay? If your observed test statistic, right, exceeds the critical value, then you conclude that you can reject the null, and your test is statistically significant, right? Statistically significant. Doesn't necessarily mean you have biological significance, but you have statistical significance. All right. So that's your test statistic. Where does a critical value come from? Okay. And I'll give you two examples. If you flip a coin, okay, and you have the number of heads, okay, the number of heads. Theoretically, and let's say you do it 100 times, let's make it nice and easy. All right, so we have 0 to 100. What you're going to get is, so you do this an infinite number of times. And that's where the frequentist name of the Neyman-Pearson comes from. So if you have a fair penny and you toss it an infinite number of times, what you find is exceedingly rarely will you find that you get no heads. Very rarely will you get 100% heads. And then most of the time, it'll bounce around 50. Okay. So it'll bounce around 50, but not every time. So you'll get 51 heads, 54, 60, 23, right? 40, 50, 51, 17, 89. And when you do this an infinite number of times, you should get a distribution like this, okay? Where you get 50, piles up on 50. So that is the null, okay? That you have a fair penny. Because that's what the Neyman-Pearson is. You're actually comparing the observed, okay, versus a critical value. And the critical value, okay, is generated from the null hypothesis. And what you're gonna do is have a distribution of expected values, figure out what your critical value is, and we'll talk about where that comes from, okay? So you have a critical value. And so if your observed comes out here, you can reject the null, okay? But here's the thing. Let's say that our critical value for 50 is, we're going to say just 95. I don't know if that's true. And then 5% of the time you'll get above 95 heads, okay? Above 95, so 96, 97, 98, 99, 100. So a value more extreme, okay? You can get that, right? It'll only occur 5% of the time that you will flip a fair penny and get this. However, if you don't know if it's fair and you're testing to see if it's a bad penny, that you'll reject that it's a fair penny 5% of the time. If you do this a number of times, right? But there's a 5% chance, okay? So that's about 1 out of 20 times you flip a fair penny, you'll conclude, right, if you do a chi-square, that you have a bad penny. All right. This critical value is this area here. Now, mind you, this is from a fair penny, so you are making an error, right? And what that error is we're going to call that this area here alpha, which is the error rate. Right? And that's the error of making, rejecting true null. Okay? We don't know if the null is actually true or false, right? When we measure these heights of corn, you don't know if it's really true or false, but what you know is how probable your observed data are given the null was true. So we can calculate the distribution of the null, okay? And that 5% is our error rate. So if we get, say, 97 heads, we're going to conclude that we have a bad penny, even though there's a small probability of getting that. That's our error rate, okay? So we don't know if the penny is actually bad or good. We can only use statistics to tell us, right? And understanding that we might be wrong. All right. So that's a penny flip. For the corn, it's a little bit different. What you do is it'll look like this. You have your distribution of, say, your control. And you look at the amount of overlap. Okay. So if this area here, okay, if this area under this curve overlaps with this, okay, less than 0.05, we can reject our null. Reject null. So if you could imagine, give you two scenarios. So let's say there's no overlap. Okay, so here's our control and our plus fertilizer, right? And here's our heights, right? So this is the frequency of heights, right? And there's no overlap. We should be fairly confident rejecting that these two are the same. So the null hypothesis is a hypothesis that, at least in this case, that you're pulling from the same population. So if you have 100 plants and 100 plants, it's very unlikely that these two populations are actually the same population, right? if you pulled 100 plants and they were from the same population, you might just happen to grab a few large ones, and what you might get is something like this. Okay, and here's the means, and the question is, are these means different enough that you say these are from two different populations, or is it still from the same population? Okay, and so you're much less likely to reject the null. If you do not reject, you simply say you fail to reject. Okay, so you don't say you accept the null, you simply fail to reject. All right, so we covered critical value, that it's generated from the null hypothesis, and that's how you generate expected values. You have an error rate of falsely rejecting a true null, okay? And usually our error rate, sort of the universal in biology, is 0.05, right? So that's the error rate we accept. And if we get a value smaller than this, we have statistical significance, okay? All right, and if it's less than that, we reject null, and we have significant data. If it's greater than 0.05, oops, we fail to reject. Okay, and then we say our data are non-significant. Insignificant data means our data doesn't have meaning, but non-significant means we're comparing it to significant data. So there's a little bit of context you need to provide. You may feel insignificant because your results are non-significant. So just keep that in mind. All right, so last thing I want to cover here. Let's erase all this. The p-value. So everything essentially comes down to a p-value. Here's what I want you to know about p-values. Ron Fisher came up with it around the 1930s and 1940s, and it was simply a rule of thumb, okay? And what happened was Ron Fisher became a giant in statistics and genetics about this time. And if you're a giant, and what I mean is an intellectual giant, you're the person publishing the papers, you're the person that people are listening to, that your rules of thumb become basically the standard, right? Not just like, here's a good idea, but here's the idea. And he actually never meant for it to become this thing. 0.05 is arbitrary. Right? It's totally arbitrary. He could have said use 0.06, 0.04, 0.01. He chose 0.05. It's just a completely, it has no meaning, right? It has, so when you say statistical significance, that doesn't mean it your data has some sort of biological significance, and it also means if you fail to reject, that your data doesn't have biological significance. You have to get that in your head that this 0.05 is just a statistical way of testing data. It's not a way of biologically testing your data, right? So the way to understand your data is through graphing and understanding what p-values are and effect sizes. And we'll get to all that. All right. So p-values, they were a rule of thumb. He just picked it as an arbitrary number. He thought it was a good one, probably just easier to remember than 0.04725. 0.05 is easy to remember. Nothing special about it. Okay. P-values. What drives p-values? Effect sizes. Right? So I already gave this with your corn plants, right? So if this is height, I'm sorry. Yeah, this is height. And so they're taller this way. You have a distribution of plants that look like this. And distribution of plants look like this. This is your effect size. So this will have a smaller p than, say, another experiment where you get this. Because now here's your effect size. Okay. So this is dealing with two plants. If you had, or examining trends, this would not be a large effect size. This would be large effect size. So if it was caffeine and heart rate, and this was your slope, it means you can drink a lot of coffee and your heart rate will increase, but not by much. If this is the relationship between heart rate and caffeine, it means you drink a little bit, and every little bit more you drink, your heart rate goes up and up and up, right? So this is effect size. So this one would have a smaller p, this would have a larger p, right? And smaller and larger relative to 0.05. So this might be smaller than 0.05, this might be greater than 0.05. So effect size is important, and that we can kind of visualize in graphs. All right. The other thing is variance. So this is the noise, okay? And effect size is the signal. All right. So what do I mean by noise? So let's look at our corn plants again. I'll give you two scenarios. Okay. We'll make the effect size the same. Okay, but this time we're going to increase variance. So let's say we have very little variance. Okay, so you can see the overlap is here, but here's your effect size. It's the same over here, but what we did was we increased the variance. Okay, so when you have small variance, you can detect, right? It's very clear that if you sample this population here, nearly every single individual will be shorter than this individual over here. Here, there's a lot of overlap, which means you can sample control plants, and it'll be much taller than many of the fertilizer plants. So the larger the variance, the larger the p-value. So it's inverse for effect size and it's relative with variance. So the larger the variance, the larger the p-value. All right, and then the third thing is degrees of freedom. Okay, degrees of freedom is related to variance. Okay, so you can have two graphs, and degrees of freedom, by the way, is the number of observations that can wiggle. And what I mean by that is those numbers can take on any value, and a certain number of observations will have fixed values. And what I mean by that is if you flipped a coin and I told you the number heads, you automatically know the number of tails. Okay. So the number of heads is free and the number of tails is fixed, right? Or you can flip that around. So for something that can take on two values, degrees of freedom is 1. For like say a dihybrid cross, right? With the 9:3 to 3:1 ratios, right? I could tell you how many yellow smooth, yellow wrinkled, purple smooth, and then you can tell me how many purple wrinkled out of, if I told you there were 200, right? So you just add up those 3 categories, you can tell me the 4th, right? But what this means is it's related to sample size, and as you increase sample size, you get more power. And so if I told you this was the distribution based on 10 plants, maybe I just chose 2 or 3 large ones here, and I happen to get a very small one over here, and it pulled those means apart, right? Just based on a random sample. If I told you this distribution and this distribution here were based on 1,000 or 10,000 plants in each population, you'd be much more confident that this effect is real, okay? So degrees of freedom, sample size goes into calculating p-value, okay? And these things balance themselves out. So if you have a small effect size, you can still get statistically significant results if your variance is small, or if you have very large samples, or a combination of both. You can have a large effect size, but can still be insignificant if you have a small sample size and large variance. So think about these 3 things as balancing each other out, or working together to make observations statistically significant or not. I think I covered everything. And so ends part 1. So tune back in, and you can get part 2. All right, thanks for listening.